Good afternoon, students. In the last lecture, we were discussing about the modern technique of plant breeding, and that is mutation breeding. In the mutation breeding, up till now we have discussed what is meant by the mutation, then the types of the mutation, and also we have discussed about the different types of the mutagens which are used in the mutation breeding. Okay, mutations, these are the sudden heritable changes which are arised other than Mendelian segregation and the recombination in the organism, and these are called as the mutation. Okay, and the mutation breeding means the creation of mutation at will and the production of the new superior varieties is called as the mutation breeding. Okay, we have discussed up now the types of the mutations and also the different types of the mutagens. For example, we have discussed the ionizing radiations, then non-ionizing radiations, and also the different types of the chemical mutagens. Okay, mutagens, these are nothing but a certain type of the substances which are used to induce the mutations, and these are called as the mutagens. Okay, mutagens, these are classified into the two groups. The first one, the radiations, and the second one, that is these are the chemical mutagens. Okay, in today's lecture we are going to discuss how exactly the mutation breeding is carried out, how exactly the mutation breeding it is practiced on the field, and the new superior variety is released. Okay, here the field technique which is practiced in the mutation breeding. Okay, there are certain important factors in the field technique, like the first one is the selection of the variety. Okay, selection of the variety which is selected for the mutation breeding, it should be the best variety available in the crops. Okay, the variety which is selected for the mutation breeding, it should be the best variety available in the crops. Then another important thing, the genetic information and the radio sensitivity of that particular variety, it should be known previously. Okay, the genetic information and radio sensitivity should be known. Then the population must be large enough as the frequency of the desirable mutation are so small. Okay, as the frequency of the desirable mutations are so small, the population must be large, so the plant breeder can detect the desirable mutations, or we can say the plant breeder can observe the large number of the population, can easily detect the different types of the mutants with the desirable characters, and that's why the population must be large enough so that the frequency of the desirable mutations are so small. Okay, frequency of the desirable mutations are so small so that they are not likely to be noticed in the small number of the population. If the very less number of plants are selected, then it is very difficult to notice the mutants with the desirable combination of the character, and that's why the population must be large enough to frequency of the desirable mutations are so small. Okay, then another important thing, the amount of plant material to be used, amount of plant material to be used depending upon the funds and facilities available and the objectives of the breeding program. Okay, as far as the it is possible, the large number of the population are selected, but the number or the amount of plant material it is selected, it is completely depending upon the funds and the facilities available and the objectives of the breeding program. Okay, objectives of the breeding program. Then the another important thing in the field technique is the part of the plant which is to be used for the mutation breeding. Okay, here in this photograph you can see the different plant parts like the seeds, then the pollen grains, then vegetative propagules like the corns, bulbs. These are used the mutation breeding. Even the complete plant, it is treated with the mutagens. Okay, different plant parts, these are used in the mutation breeding, like the seeds, seedlings, then the vegetative cuttings, then the entire plant also it can be used for the mutation breeding. Okay, the plant part to be used, it is depending upon the objective of the plant breeding. For example, if the plant it is to be used in the hybridization, okay, if the plant it is to be used in the hybridization, then generally the pollen grains, that is, these are treated with the different mutagens. Okay, these are treated with the mutagens. Okay, and generally the vegetative propagules, corns and the bulbs, these or the cuttings, generally these are used in the fruit trees. Okay, the vegetative propagules like the corns, bulbs or the cuttings, these are generally used in the mutation breeding of the fruit trees. Okay, then the another important thing while using the seeds there are, that is, for example, the aged and the soaked seeds. Aged and the soaked seeds show the greater frequency of the induced mutation than the fresh air dried seeds. Okay, aged and the soaked seeds show the greater frequency of induced mutations than those of the fresh air dried seeds. And it must be remembered that the seeds should not be too old or these should not be too young. Okay, then the water content or the moisture content in the seeds, it should be proper, it should be appropriate. Okay, it should be proper and it should be appropriate, and that's why the seeds which are having the appropriate amount of moisture, they show the higher radio sensitivity. Okay, they show the higher radio sensitivity. Sometimes the seedlings, the small plants, these are also used to give the treatments. Okay, because the seedlings these are small sized, these are easy for transportation, and these are also easy to handle. Okay, the seeds of the plants, these are used for the treatment. Then also the seedlings, these are also used to give the treatment of mutations, because the seedlings, these are the small sized plants, these are easy for transportation, and these are easy to handle. Okay, sometimes the completely developed plant, it is also used to give the treatment of different mutagens. Okay, generally in completely developed plants, the treatment it is given at the time of flowering. Okay, generally whenever the complete plant it is used to give the treatment of different mutagens, generally the treatment it is given at the time of the flowering, because the meiotic cells, these are more sensitive than the mitotic cells, and that's why generally whenever the complete plant it is used to give the treatment, then generally the treatment it is given at the flowering stage, because the meiotic cells are more sensitive than the mitotic cells. Okay, that is about the different plant parts which are used in the mutation breeding. Okay, so sometimes the seeds of the plants, these are used to give the treatment, then pollen grains, then different vegetative propagules like the corns, bulbs, or even the complete plant, it is used to give the treatment with the different types of the mutagens. Okay, then the next one, the another important thing in this is the treatment. Okay, we have discussed the different types of the mutagens. Mutagens, these are the substances which are inducing the mutations in the living organism. Okay, we have discussed the radiations. In the radiations also we have discussed the ionizing radiations and the non-ionizing radiations. Okay, then while discussing in the different types of the mutagens, we have also concluded that the use of X-ray and the gamma ray, it is most common method which is used to give the treatment. Okay, use of X-ray and the gamma rays, this is the most common method which is used in the mutation breeding. Okay, before giving the treatment, it is very essential to decide the proper dose of irradiation so that the frequency of the desired mutations is increased and that of undesirable can be decreased. Okay, it is very essential to decide the proper dose of irradiation so that the frequency of desired mutations increased and that of undesirable can be decreased. Then the proper dosage, the proper dose of the treatment, it varies from crop to crop, and it should be determined for the every crop by changing the duration of the treatment and the distance between the source and the plant material. Okay, it is very essential before practicing the mutation breeding in the field to determine the proper dose of the treatment for every crop. Okay, because the radio sensitivity of the every crop, it is very different. Okay, the it is, so that it is very essential to determine the proper dose of, for which is required for the every crop, so that the frequency of the desired mutation, it will increase and the frequency of the undesirable mutation will get decreased. Okay, and how the this proper dose of treatment, proper dose of the different mutagens, it is determined. It is very simple. It is determined just simply changing the duration of the treatment and changing the distance between the source and the plant material. Okay, we have discussed the we have already discussed the different methods, of which the x-ray and the gamma rays, these are the most common methods which are used for the treatment. Okay, apart from these, the different types of the chemicals, these are also used to treat the plant material. Okay, that is about the treatment. Okay, then here after the selection of the plant material, then they giving the treatment with the proper dose to the plant material. Okay, then there is a actual field work starts. Okay, actual field work starts, that is the seeds which are given the treatment with the proper dose of the different mutagens, that is these are thoroughly washed and these are then sown in the field along with the mother strain for the comparison. Okay, here the mother strain, it is also sown along with the treated seeds, that is just for the comparison. That is these mother strain, we generally called them as the check. Okay, the seeds which are treated, these are thoroughly washed and these are sown in the field along with the mother strain for the comparison. These mother strains, these are used as the checks. Okay, the seeds, as these are treated with the different types of the mutagens, the treatment, it may produce the visual disturbances. It may produce the visual disturbances like death the death. If the due to the treatment of the mutagens, the death of the plant takes place, then there nothing remains for the further study. If the death of the plant takes place, then nothing remains for the further study. Sometimes it may be responsible for the growth inhibition and the morphological and the developmental abnormalities. Okay, these growth inhibition and the morphological and developmental abnormalities, these may be or may not be the potential interest to the plant breeder, as these are temporary and plants can recover from such abnormalities. Okay, the growth inhibition and the morphological and developmental abnormalities, that is these are temporary and the plants can recover. That's why the plant breeders are not interested in these three visual disturbances like the death, growth inhibition and morphological and developmental abnormalities. Plant breeders, these are interested only in the genetic changes. Okay, these are only interest, they are only interesting in the genetic changes, because the genetic changes, these are very rare. These are very rare, these are very useful in the crop improvement. Okay, these are very rare and these are useful in the crop improvement. Then the gene mutants, the mutants, they differ from their parental strain in any character. Okay, the first generation which is in the field, it may differ. These are the mutants which are differing from their parental strain in the external or even in the internal character. Okay, even in internal character, these changes which occurred due to the treatment, they may be morphological. There may be the changes in the phenotypic characters, or there may be the changes in the physiological. Okay, the changes may be morphological, these may be physiological, or these may be biochemical. Okay, the changes which are occurred due to the treatment, they may be morphological, physiological, or the biological. Another important thing, these changes, they may immediately appear in the first radiated generation, or they may not be get expressed in the first generation. Okay, these changes which immediately may or may not get expressed phenotypically in the first irradiated generation, which is referred as the R1 generation or the M1 generations. Okay, that means the mutations which takes place, these may be recessive or the dominant. Okay, the mutants, they may be recessive or the dominant. Okay, if the mutations are dominant, they will readily appear in the first irradiated generation. Okay, but generally it is very common that the mutations are recessive. Okay, that is they not get expressed until and unless the two recessive genes comes together. Okay, when only the two recessive genes comes together, only at that time the character will get expressed. Okay, and that's why whenever that is the mutations are recessive, it will require the intervention of the one or more generation after their occurrence. Okay, it will require one or generation after their occurrence. That means it is not necessary that all the characters which are completely different from their parental state, they get expressed in first generation. It may be possible that these characters may get expressed in second or the third generation, as these characters, as these mutations are recessive. Okay, and that's why each and every suspected plant in the first irradiated generations are picked, and these are kept separate. Okay, how the selection is carried out, each and every suspected plant, it is picked and it is kept separate. Okay, and another important thing in the first irradiated generation is that the plants should be allowed to cross pollinate. Okay, the plants should be allowed to cross pollinate. Okay, here, no, here I have mistakenly written it that the plants, you should read it as a plants should not be allowed to cross pollinate. Okay, the plants should not be allowed to cross pollinate. Okay, that's why the selection, how the selection is carried out, the almost each and every suspected plant is picked and kept separate. Okay, in the first generation R1 or the M1 generation. Then in the R2 generations, the seeds which are collected from the R1 plants, these are grown in the separate individual rows to obtain the R2 generations. Okay, the seeds of each of the plant, which seeds of the each of the plant in R1 generation, that is these are sown in the separate individual rows to obtain the R2 generations. Okay, then here in the R2 generations, the screening and the selection of the irradiated material, it is started in the R2 generation rather than the R1 generation. Okay, in the R1 generation, what happened, each and every suspected plant is selected. Okay, because it is not necessary that all the mutations, that is they get expressed into the first generation, and that's why, that is the reason why the screening and the selection of irradiated material, it is started in R2 generation rather than the R1 generations. Okay, in the R2 generations, the plants with the desirable characters are selected, these are harvested, they are threshed, and the seeds which are collected, these are kept separately. Okay, the seeds of each of the plant, that these are kept separately. Okay, the plants with the desirable characters are selected, these are harvested, these are threshed, and the seeds of the each of the plant, these are kept separately, these are not mixed with each other. Okay, these are the activities which are carried out in the R2 generation. Then in the R3 to R5 generations, the same procedure like the R2 generation, it is repeated in the R3 to R5 generation. That is the seeds of the plants with the desirable characters. These are the desirable plants, or we can say the plants with the desirable characters are selected. These are harvested, then their seeds are kept separately, these are then sown in the field to obtain the next generation. The same procedure it is repeated in the R3 to R5. The best ones are selected and the others are discarded. Okay, then later on the main yield trials are carried out. Okay, main yield trials, these main yield trials, how the main yield trials are carried out? The highly useful mutants, or we can say the highly useful mutants with the desirable characters, these are selected from the R5 generation. Okay, here you can see the highly useful mutants, these are selected from the R5 generation. Then the yield trials are carried out in the R6 and the R7 generations. Okay, yield trials are carried out in the R6 and the R7 generation, and simultaneously the regional yield trials, or we can say the multi-locational yield trials are carried out. Okay, that is just to find out their wider adaptability, whether these strains, whether these mutants are adaptable to the different environmental conditions or not. In parallel, simultaneous trials are also carried out. Okay, then the performance of all these strains, these are then the performance of the all these strain, it is compared by the plant breeder. The strains which are superior in the desired character, these are released as the new variety. Okay, the performance of each of the strain, it is then given to the plant breeder, and then plant breeder decides the superior strain and then release this superior strain as the new variety. Okay, then here the time which is required to release the new variety by using this technique. Okay, generally the time required to release the new variety by using the mutation breeding technique, it varies from problem to problem and from crop to crop. Okay, and generally it is depending upon the nature of improvement which is needed. Okay, it is depending upon the nature of improvement which is needed and the stage at which it is obtained. But when we compare the time which is required in the mutation breeding to release the new variety with the other conventional breeding technique, then it is usually less. Okay, the mutation breeding takes usually less time when compared with the conventional breeding technique to release the new variety. Okay, that is about the field technique and the time which is required to release the new variety in the mutation breeding. Okay, here you can see the entire process in a one photograph, entire field technique in the one photograph. Okay, here you can see the M1 generation, the seeds which are treated with the different mutagens. Here you can see the mutagen treated seeds, these are space planted. The seeds from the individual plants are harvested separately. Okay, in the M1, each and every suspected plant is selected, and there these are harvested, their seeds are collected, and these are kept separately. Here you can see the individual plant progenies are grown. Okay, here the individual plant progenies are grown. And from the M2 generation, the fertile, vigorous, normal looking plants, these are harvested separately, their seeds are kept separately. Okay, in the M3 generation, here you can see the individual plant progenies from the selected plants are grown. Okay, from the selected plants are grown. Then the superior plants are selected from the superior progeny, showing the segregation. Okay, and homogeneous mutant progenies may be harvested in the bulk. Homogeneous mutant progenies means the plants which are showing the similar characters, similar phenotypic characters. These may be selected and these may be bulked together. Then here in M4 generation, the individual plant progenies from the selected plants, these are grown. Okay, these are grown, and the superior homogeneous lines are harvested in the bulk. Okay, in the M5 generation, here you can see the preliminary yield trial with the suitable check. Preliminary yield trials are carried out in the M5 generation, and superior lines from the M5 are selected. And here you can see from the M6 to the M8 generation, the multi-location yield trials are carried out in the regional experimental stations. Okay, and then their performance, it is reported to the plant breeder, and then the outstanding lines or the superior varieties, these are released as the new variety. The seeds of these new variety, these are then multiplied in the ninth generation, M9 generation. The seeds are multiplied and these are then provided to the farmers for the commercial cultivation. Okay, that is the whole process of the mutation breeding. Okay, which is shown here in just only one photograph. Okay, then the precautions which are generally taken while practicing the mutation breedings. Okay, here the mutants which are isolated, these are really due to the mutations and not due to the intermixing, crossing, or the segregation. Okay, that is sometimes while practicing the mutation breeding, it may happen that the due to the intermixing or due to the crossing or the segregation, the variation may be created. Okay, but here the plant breeders are only concerned with the variations which are arised due to the mutation treatment. Okay, that's why the mutants isolated, they must be really due to the mutations and not due to the intermixing, crossing, or the segregation. Every other possibility of mode of origin of variation in the individuals must be excluded from the mutant population. Okay, every other possibility of mode of origin of variation in individuals must be excluded from the mutant population. Okay, and that's why the so that the variation which appears in the plants, it it is only due to the mutations and it will be not due to the intermixing, crossing, or the segregation. Okay, and another important thing is that to complete the process, to carry out all the field practice, the plant breeder must be well trained in the breeding technique, and he must be very keen observer. Okay, he must be very keen observer so that he can pick up a slightest deviation in the treated population. Okay, he must be very keen observer so that he can pick the slightest variation in the treated population and include his selection for the further investigation. Okay, so that here we are saying that the plant breeder must be well trained in the breeding technique and he must be very keen observer. Okay, then the applications. Okay, utilization and the applications in the self pollinated crops. The self-pollinated crops, as these are genetically homozygous, as you know, that is the self-pollinated crops, these are naturally homozygous. The improvement, it is not possible until and unless some variation is either present naturally or the created artificially. Okay, genetic variation, it is the basis of any hybridization, any plant breeding program. Okay, if the genetic variation is not present, then it is not possible to improve the crop and to release the new variety. Okay, but the self pollinated crops, these are naturally homozygous. Okay, and that's why here, as these are homozygous, even a slightest variation in their character, it can be detected easily. Okay, if we are using the mutation breeding in the self pollinated crops, then it is very easy to observe the different types of the variations which are arised due to the mutation breeding. Okay, and therefore the mutation breeding, it is used widely and extensively in the self-pollinated crops, as the mutants can be easily observed and these can be separated from the other plants, and that's why the mutation breeding, it is used extensively in the self-pollinated crops. Okay, but in the cross-pollinated crop, as you know, that is the cross-pollinated crops, that is these are heterozygous. Okay, the as these are naturally heterozygous, the genetic variability, it is in abundance in naturally. Okay, these cross-pollinated crops are naturally genetic variables. Okay, and that's why they give the different types of the progeny. Okay, they will give the different types of the progeny, and that's why it will hamper the detection of the different types of the mutants in the cross pollinated crops. Okay, as these are naturally heterozygous, various types of the progenies are produced, and that's why it will become very difficult to observe the different types of the mutants, to select the different types of the mutants in the cross pollinated crops. And some of the plant breeder, that is, they believe that as the cross pollinated crops, these are naturally genetic variable, it is completely unnecessary to use the mutation breeding technique in the cross pollinated crop. Okay, mutation breeding technique, it is simply creating the different types of the variations in the plants, and just from these variations the plants with the desirable characters are selected. Okay, that is the mutation breeding technique. But in the cross pollinated crop, the naturally the these crops, these are genetically variable. Okay, that's why it is completely unnecessary to use this method for the improvement of the cross pollinated crops. Okay, some of the plant breeder believe that as these are naturally genetic variable, it is completely unnecessary to use the mutation breeding technique in the cross pollinated crop. Okay, then the application of the mutation breeding in the vegetatively propagated plants. Vegetatively propagated plants, these are highly heterozygous. Okay, these are highly heterozygous and they hardly permits the purposeful improvement by the conventional breeding techniques. Okay, then these the sexual reproduction in these vegetatively propagated plant destroys the all the valuable characters. Okay, and therefore plant breeder believe that the it is advantageous to use the mutation breeding technique in the vegetatively propagated plants. Okay, that is all about the application of the mutation breeding in the self pollinated crops, cross pollinated crops, and in vegetatively propagated plants. Okay, then there are certain limitations for use of this method. Okay, that is, this method cannot be used until and unless the genetic information, there is a full information about the genotype of material, then proper mutagen, its efficiency, frequency, then it cannot be used. Okay, in absence of the full information about the genotype of plant material, then proper mutagen, its efficiency, then frequency, it cannot be practiced in the improvement of the crops. Okay, then here another limitation to use this method, that is the irradiation facilities and trained personals, these are available only in few institutes. Okay, these are available only few institutes. Okay, for example, we have discussed the different types of mutagens like the neutrons. Okay, the treatment, it can be only given in the atomic energy departments which are established by the governments. Okay, and such institutes are very few. That's why we are seeing here, that is the irradiation facilities and the trained personals are available only in few institutes. Okay, then this method, it does not produce the ready-made varieties, it does not give the ready-made varieties. This method only produces inexhaustible variations. Okay, it produces only the inexhaustible variations, and the desired ones are picked. The desired ones are picked, and these are then used directly or the indirectly to release the new variety. Okay, these are the limitations of the mutation breeding. Okay, the generally when the improvement by using the conventional breeding are available, when the improvement by the conventional breeding can be done, can be possible, so no one can jump directly for the mutation breeding to release the new variety. Okay, always remember that if the improvement in the crops, it is possible by the conventional breeding techniques. For example, plant introduction is there, then selection techniques are there, then the we have discussed the hybridization and the heteros. If the improvement is possible by using these methods, then no one can jump to the mutation breeding. And if the mutation, if the improvement is not possible by using this method, then only in that case the plant breeder use the mutation breeding technique to improve the crop and to release the new variety. Okay, that is all about the mutation breeding. That is all about the field technique which is used in the mutation breeding.